Hello students, this is a video class of half an hour duration in which we will deal glycogen metabolism. This first video will have two modules in it. The first module will be glycogen breakdown or glycogenolysis, and the second module glycogen synthesis or glycogenesis. And we'll have another video of third module which will deal the regulation of glycogen metabolism. Now, glycogen, as we know, it's a polysaccharide of glucose subunits which are linked together through alpha 1,4 linkages or bonds. So that's a linear chain, but of course there will be side branches which comes after, let us say, 8 to 12 subunits of alpha 1,4 linkage. There can be one side branch in alpha 1,6 bonding. So that's how glycogen is formed. And this is much more branched when we compare with another polysaccharide, namely starch. This is much more branched when comparison to the structure of starch. Now, glycogen can be seen in cells like granules, as we see here, primarily in liver and muscle cells, as well as in some other cells also, like brain cells. It is seen not much concentrated like this, as we see in liver and muscle. So that is glycogen metabolism. Let's go through the details of glycogen metabolism. Now, glycogen is the best readily mobilizable major storage form of glucose. It is insoluble and so osmotically inactive, which means if it is present in the cell, it cannot escape from that cell, and cell can use it now or later. And another advantage of storing glucose as glycogen is that it provides higher energy than same weight of glucose if it is stored as glucose. Now, there is a comparison chart here of liver and muscle glycogen. Liver concentrates glycogen because we can see here 5% is the liver tissue's glycogen, whereas in muscle it's just 0.7% only. But obviously muscle mass is greater than the weight of liver, and as a result, liver stores 90 grams of glycogen in it, whereas muscle can store up to 245 grams of glycogen in it. And the reasons for storing glycogen as fuel reserve, we can go through it. Readily mobilizable form of glucose, it can be obtained instantaneously. It helps anaerobic conditions also, as obviously in muscle cells, it helps anaerobic respiration also. Now basically, when we have food, the glucose available from food will last for around 5 hours, after which glycogen will be broken down. And the glycogen reserve can be used up to 18 hours. So that is the advantage of storing glycogen as fuel reserve. And later, when we continue fasting, the body will turn to fats and fatty acids for energy or ATP production. Now, this is a structure, basic structure of glycogen, as we saw, or as you know. This is the alpha 1,4 linkage, alpha 1,4. This is alpha 1,6 linkage, and we can see a reducing end here because the anomeric carbon, the first carbon, is there, which is having the aldehyde group. Though this is the first carbon, it's a reducing end, and these are the non-reducing ends. And these non-reducing ends are important because both in glycogen breakdown and glycogen synthesis, these non-reducing ends are important. So just keep it in mind that when we say when I mention non-reducing end, I mean this end of the glycogen. Yeah, this is another picture which shows the branching and alpha 1,6 linkage. Glycogen function. Now, function obviously in liver and muscle, it has got slightly different functions. In liver, what liver does is that the synthesis and breakdown of glycogen is regulated in liver to maintain blood glucose level. So the glycogen stored in liver is actually distributed throughout body when it is broken down to glucose. Whereas in muscle, what happens is that the synthesis and breakdown of glycogen is regulated to meet the energy requirements of muscle cell itself. So in muscle, the glucose produced by glycogen breakdown is used in muscle cell itself. So liver is doing a sort of a charity function. It's a charity organ in our body, and as we know, liver is doing a number of functions, and one of them is glycogen storage and supply. Now glycogen, before we go on to see the actual breakdown of glycogen, this is glycogen when broken down will give glucose 1-phosphate, then glucose 6-phosphate, and this glucose 6-phosphate is a junction, a metabolic junction from where we can go to glycolysis, or we can produce glucose and can be distributed to cells, as happens in liver. Or the glucose 6-phosphate can also be utilized for PPP, that is hexose monophosphate pathway, HMP pathway or PPP pathway, to produce ribose and NADPH. And we move on to the first module, glycogenolysis. Now glycogenolysis is the breakdown of glycogen, and the glycogenolysis is a catabolic process, obviously, and it is never a reversal of glycogenesis. Now glycogenesis is the synthesis of glycogen. There are separate pathways. There are separate pathways for glycogenolysis and glycogenesis, and this is the case most usually in biochemical reactions or metabolic reactions. For example, fatty acid synthesis is not the reversal of fatty acid breakdown. This takes place in cytoplasm, and glucose is released from non-reducing end of glycogen. So you have to note here, non-reducing end, that we have already discussed. There are certain areas or positions in glycogen structure where we can see non-reducing ends. So the enzymes actually start to attack from non-reducing ends of glycogen. Now, this is the most important, most important slide in the context of glycogenolysis, glycogen breakdown, because it represents a whole lot of reactions involved in glycogenolysis. There are 3 enzymes involved. The most crucial or the rate limiting enzyme is the first enzyme, namely glycogen phosphorylase. Then there are 2 more enzymes, one for glucan transferase, alpha-1 for glucan transferase, and alpha-1,6 glucosidase, 3 enzymes. And the most important one is the phosphorylase enzyme. Now, we have represented a chain here, a part of the glycogen molecule here. It is attached, obviously, the rest of the structure, the core structure. And it's been given in different colors also. The subunits have been given in different colors also, so as to make the process of learning more easier. Now, what a phosphorylase does is that it actually undertakes phosphorylatic cleavage of glycogen. No, it is phosphorylatic cleavage and not a hydrolytic cleavage. Hydrolytic cleavage involves split of or hydrolysis of water molecules. But here, in phosphorylatic cleavage, it involves a phosphate group. See here, an inorganic phosphate is given. And there are 8 molecules from non-reducing end, like this. So, each of them are cut away, like this. Each of them are cut away, or the alpha-1,4 linkage is cut away by glycogen phosphorylase, so as to produce not glucose, but glucose 1-phosphate. Glucose 1-phosphate. So this is the molecule, this is the result of phosphorylase action. And phosphorylase is unique in that it can cut alpha-1,4 only, and also it can cut alpha-1,4 till it reaches a particular molecule which is away from 4 molecules till it reaches 4 molecules, 4 glucose molecules away from a side branch. You can see 3 blue colored molecules or subunits and one green colored subunit here, and it is attached to alpha-1,6. So phosphorylase will cut alpha-1,4 subunits till it reaches till its action reaches 4 subunits away from a side branch point, like this. Okay, here also it is a side branch point. So from where the yellow colored unit starts. Okay, now another enzyme. So what is the result? This is the result. This is the result of phosphorylase action. It is attached to the rest of the molecule. And from there on, another enzyme takes charge. It is alpha-1,4-glucan transferase. And what alpha-1,4-glucan transferase, as the name indicates, it is a transfer. It is an enzyme which helps transfer of a bunch of glucose molecules. Here it is a trisaccharide, or 3 glycosyl molecules are shifted as such to this position, so as to make the chain look like this. So this 3 molecules, and not the fourth molecule remains there attached to the original chain in alpha-1,6 mode. So this is what alpha-1,4-glucan transferase does. It just shifts only, but phosphorylatic or hydrolytic cleavage is not involved. Now, this phosphorylase enzyme, you can just guess that it can go on cutting from here, because these 3 blue colored molecules can be removed till it reaches here, because the action, as I said earlier, it can release glucose 1-phosphate molecules till it reaches 4 molecules away from a side branch point. So these 3 molecules can be cut away by phosphorylase. It can carry on with its function, but not to here. At that time, to solve this problem, another enzyme comes into picture, that is alpha-1,6-glucosidase, which is also called a debranching enzyme, alpha-1,6-glucosidase. It actually cuts away that single stump, that glucose molecules. It is single glucose molecules, that green colored subunit, which is attached to the parent chain through an alpha-1,6 linkage. So alpha-1,6-glucosidase is specific for cutting alpha-1,6 side branch unit, and this is actually not phosphorylatic cleavage, it's a hydrolytic cleavage. You can see a water molecule is used, and the product is that product is not having an attached phosphate in it as a result. And what is the product? It's just a glucose unit, assembled glucose units. So rest of the molecules, wherever it is released, it is released as glucose 1-phosphate, except for this particular one molecule that is attached in alpha-1,6 mode. It will be released as glucose as such. Now, this is another representation. Phosphorylase acts till it reaches 4 molecules away, and of the 4 molecules, 3 molecules will be shifted like this. It will be shifted to here, and the rest of the molecule is this one, that green one, that will be released as glucose. And from here it can be released by phosphorylase. Glycogen phosphorylase, the most crucial first enzyme. Now, 3 enzymes are involved, as we saw. Glycogen phosphorylase, the most crucial enzyme, rate limiting enzyme. The second one is alpha-1,4-glucan transferase. The third one is alpha-1,6-glucosidase, or the debranching enzyme. Glycogen phosphorylase, this is the actual work carried out by glycogen phosphorylase. Obviously, it acts on the non-reducing end of glycogen, thereby releasing glucose 1-phosphate, producing glucose 1-phosphate, and it reduces actually glycogen by 1 unit. Because that 1 unit is released as glucose 1-phosphate, and this particular unit becomes the new non-reducing end, from where it can again attack here, this point, till it reaches 4 units away from a side branch. Rate limiting step, present in liver and muscle. Alpha-1,4 linkage is the one attacked till it reaches 4 units. This one, 4 units away from 1,6 branch point. So that is the point to be remembered here. It attacks not from from a single non-reducing end. The enzyme attacks wherever non-reducing ends are there. This particular enzyme will start to attack and release single glucose 1-phosphate molecules. Now, consider a case when glycogen phosphorylase enzyme alone is acting. What happens is that you will get a highly branched structure with the alpha-1,6 attached to the parent molecule or original molecule. So this type of structure will be called limit dextrin. But upon this, the further action of enzymes will take place Now, first of all, the shifting of a trisaccharide unit will be done by this particular enzyme, glucan transferase, and the debranching of that single alpha-1,6 molecule will be done by alpha-1,6 glucosidase, the debranching enzyme. It is the result of the concerted action of these three enzymes that we get, what is it? Glucose 1-phosphate. So glycogen phosphorylase, glucose 1-phosphate, and that will be instantaneously converted to glucose 6-phosphate in cells with the help of the enzyme phosphoglucomutase, another enzyme, phosphoglucomutase, because glucose 6 conversion to glucose 6-phosphate is important in some cells, because that's how we can keep the glucose molecule confined to the those cells. Okay. And what is the fate of glucose 6-phosphate? It depends on different organs. What happens in liver is that, and liver cells are unique in that liver cells will have this glucose 6-phosphatase enzyme. This is unlike muscle or let us say brain cells where this particular enzyme is absent. With the help of this or the presence of this enzyme in liver helps liver to convert glucose 6-phosphate to glucose. So obviously when glucose units are there inside the cell, it can easily cross the membrane of the cell, as happens in liver. So the released glucose is transported across the liver cell membrane to reach blood circulation, and through blood circulation it reaches the whole body, the whole tissues and cells. So liver here actually acts as a charity or sort of organ, as I said earlier, because it stores glycogen, cuts it into glucose 1-phosphate, glucose 6-phosphate, then stores as glucose 6-phosphate, then glucose is produced and released to tissues, to blood first, then to tissues. But this is unlike in the case of muscle, where glucose 6-phosphatase, this is absent. So what happens is that whatever glucose that is produced inside muscle cells from glycogen will remain inside muscle cell and be utilized by muscle cell itself. This is the same as in brain also. There again glucose 6-phosphatase is absent in brain. And brain, most importantly, brain and RBC, as we know, brain uses glucose as the primary energy source. And it is during prolonged starvation only that brain turns into some other fuel molecules. So it's the primary need of the brain that it should retain whatever glucose that is present inside it. So there is no glucose 6-phosphatase available in brain also. So this is the role of the liver. This is a unique role of the liver, that whatever glucose whatever glycogen is present in it, converted to glucose will be used by other tissues. So this is glycogenolysis or glycogen breakdown. Now we move on to the second module, glycogenesis, or the synthesis of glycogen, which is an anabolic process. And unlike glycogenolysis, this is an energy consuming pathway also. And obviously glycogen synthesis and glycogenolysis or the degradation are separate pathways, different pathways. It's not the reversal. Now degradation is given in brief here, and synthesis, which we are going to see, is represented here. Degradation obviously is a phosphorylatic cleavage of glycogen with release of glucose 1-phosphate, whereas synthesis involves a few more molecules like UDPG here. This is uridine diphosphate glucose. It's a carrier of glucose, activated glucose, UDPG. This is a sugar nucleotide also. And there are some other molecules also which we will see, like glycogenin during the synthesis, the process of synthesis. Now this is basically glycogen primer, which carries a few molecules of glucose in it. It accepts glucose from UDPG, and this glycogen becomes N+1. An additional glucose is added to glycogen. So that's how the glycogen primer elongates. And we saw this person here, Louis Leloir, who's a Nobel laureate in chemistry in 1970 for his discovery. He is credited with the discovery of sugar nucleotides and their role in the biosynthesis of carbohydrates. So this is a sugar nucleotide, UDPG. UDP carrying glucose is a sugar nucleotide. And the structure of this sugar nucleotide is illustrated here. Obviously, there is a glucose molecule attached to this particular structure. There is a uracil and a pentose phosphate, a ribose, which is connected with the glucose, bonded with the glucose through two phosphate groups. And this glucose is the activated, UDPG carries a glucose, which is the activated form of glucose. Just like we can say that ATP is carrying an activated phosphate, or acetyl coenzyme is carrying an activated acetyl group, like that. And UDPG is actually formed from glucose 1-phosphate with here. It actually acts with UTP, then triphosphate. Hydrolysis is involved with the release of this pyrophosphate, and what is formed is UDPG here. The enzyme involved is UDP glucose pyrophosphorylase. UDP glucose pyrophosphorylase, the enzyme involved in the process. Now obviously the energy comes from the hydrolysis of this molecule with the release of this pyrophosphate. So this is how UDPG is formed. Obviously glucose 1-phosphate is actually formed from glucose 6-phosphate, which in turn is formed from glucose or phosphorylation of glucose with expenditure of ATP there. So an expenditure of ATP is also involved in this process, and we will account for that when we calculate the stoichiometry or energy calculation at the end of glycogen synthesis. Now, another molecule I mentioned was glycogenin. It's a protein, it's an enzyme, it's a unique enzyme because it can undertake what is called autocatalysis, in that it acts as a substrate and as an enzyme of the same substrate. It's a homodimer, which means two subunits of same kind, where each of them are having 37 kilodalton molecular weight, these subunits, and they are classified as they are enzymes. They are classified as under transferases, specifically Glycosyl Transferase. And now what they do is that it's an enzyme that catalyzes the attachment of glucose molecules into its own structure. That is, this is a protein because and hence it will have amino acids in it. It is having a tyrosine unit at 194 position, and glucose will be attached to this tyrosine subunit. So this is called autocatalysis, and this step actually initiates glycogen synthesis, and glycogenin with attached glucose subunits. It forms a primer or it sets up glycogen synthesis or glycogen synthesis, glycogenesis. Now this is a representation of how glucose from UDPG is added to the tyrosine residue of glycogenin. Obviously this is given here. This is a part of glycogenin. This is tyrosine residue shown here, and this is UDPG. It attaches, this one attaches here, and bond is formed. This is called O-linked tyrosine, O-linked glucose residue, and what is left is UDP here. So the first glucose subunit is added to glycogenin, and this is followed by addition of further glucose units, and they also comes from actually the new subunit. The new glucose unit also comes from UDPG itself. So this is another molecule of UDPG that attaches to the previously attached glucose subunits by alpha-1,4 linkage here, and again a UDP is left behind here. So this is how one by one molecules, glucose molecules are attached to the original glycogenin parent molecule one by one. And actually these glucose subunits, the incoming glucose subunits attached to the hydroxyl group of tyrosine 194, as I said. The 194, the amino acid, that is a tyrosine, glucose molecule, the first glucose molecule attaches to the hydroxyl group of this tyrosine residue. And following, 7 more molecules of glucose or glucose subunits are added to it. So this is the structure, the primer formed finally. And after that, glycogen synthase, the enzyme involved in glycogen synthesis, the key enzyme or the rate limiting enzyme involved in glycogen synthesis, namely glycogen synthase, takes charge. So that this particular enzyme is involved in extending the chain from non-reducing ends. Yeah, these are non-reducing ends, as we had seen in one of the slides. There are non-reducing ends. It is actually from non-reducing ends that during glycogenolysis, individual glucose units were released. Likewise, the individual units of glucose are added to the non-reducing ends itself. And what happens to glycogenin is that the glycogenin remain covalently attached to the reducing end of glycogen. Will be still seen attached to the original or the final glycogen subunit. And since there are two chains in glycogenin, we can see here the tyrosine 194 with the attached glycogen molecule, and this is the second subunit of the dimer. Again here also tyrosine 194 with attached glycogen, the final glycogen structure there. So this is a dimer, that's why this has been made possible. And yeah, here also you can see this is actually two molecules of glycogens which still remain attached to the original glycogenin or glycogen primer. Now glycogen synthase is the molecule, is the enzyme, the key enzyme or the rate limiting enzyme involved in glycogen synthesis, glycogenesis. It actually catalyzes the elongation of glycogen chains in alpha 1,4 by adding alpha 1,4 subunits. So this is representation for that. This is the primer. As we know, there are some glucose units attached to it, and this is the non-reducing end, to which we here we have represented UDPG. And of course, it should be noted that whenever a glucose is attached, it comes or it is supplied by UDPG. So the activated glucose in UDPG is the one which is attached to the previous chain to which it gets attached. Likewise here, so the UDP, as you know here, the UDP is glucose is added to the non-reducing end. UDP is left behind, and this particular pink colored subunit comes here, and the chain is elongated. It becomes glycogen N+1 residue. Likewise, any number of glucose subunits can be added in alpha 1,4 fashion. But there is a problem. The glycogen synthase enzyme, it cannot catalyze branching of glycogen. So for branching, another enzyme is involved, just like glycogen degradation. There were different enzymes for debranching. Here also there is an enzyme for branching purpose. This is the enzyme. The name of the enzyme is alpha 1,6 transglycosidase. And this branching enzyme is very specific in its action. A branch is actually created by breaking an alpha 1,4 linkage, and this is actually broken from a linear chain, and the piece of glycosyl residues, a stretch of glycosyl residues are taken to another position where it is bonded in alpha 1,6 bonding. So this is how a new branch is formed, and there are definite branching rules. Whenever a segment of glycosyl residues are taken from a linear chain, it should have or it will have 7 residues in it. So 7 residues are taken, and it is taken from a branch which is already having at least 11 residues with a non-reducing end in it. So this is another rule. And again, when this stretch of 7 residues are taken to another position for creating a branch is created at least 4 residues away from another branch or a previous branch. So these are the rules, and we can see the illustration here. This is a straight chain, and this also is a straight chain. a segment is, it cannot be taken from here because it is not having 11 subunits in it, but it can be taken from here. So this particular stretch is having more than 11 subunits, 11 or more subunits, from which 7 subunits are removed by branching enzyme. And this 7 glycosyl residues are taken to another position, let us say here, and it is fixed through alpha 1,6 bonding. And whenever this is done, it is ensured that the enzyme ensures that there are or this new branch is at least 4 residues away from a previous branch. So this is the rule, branching rule, and this is being conducted by alpha or done by alpha 1,6 trans glycosidase. Now further extension of this chain, like this chain and this chain, can be done by glycogen synthase. So that is done. And the branches grow further by addition of alpha 1,4 glycosyl units, and further branching also can be done by our branching enzyme, this particular enzyme alpha 1,6 trans glycosidase. And the glycogen molecule thus branches further and further and further with the glycogen molecule inside it. As we know, there are it's a homodimer. So on either side, this is a glycogen molecule and this is a glycogen molecule along with Glycogen Primer, the dimer already fixed there. And we can now see the energy expenditure involved in glycogenesis, glycogen synthesis. Glycogen synthesis actually involves activation of glucose. First of all, glucose is converted to glucose 6-phosphate, for which one molecule of ATP is utilized. And thus glucose 6-phosphate further gets converted to glucose 1-phosphate, where there is no expenditure of energy, but glucose 1-phosphate is activated with expenditure of UTP, that is by addition of UDPG. So actually for each molecule of glycogen, each molecule of glucose that is added to glycogen, 2 molecules of or 2 high energy molecules are used up, one in the form of ATP and one in the form of UTP. And this is further illustrated here: glucose plus ATP, glucose 6-phosphate. Further glucose 6-phosphate gets converted to glucose 1-phosphate, and glucose 1-phosphate along with UTP, it gives UDPG. So this is how one ATP here and one UTP here are utilized, and we sums up here glucose plus 2 ATP plus glycogen n times, which means glycogen having n number of glucose units in it, and it becomes n plus 1, glycogen n plus 1. So this is the energetics of glycogen synthesis or glucose storage.